Okay. All right, so today's mini lecture is going to be on the body buffer systems. We're going to talk about two. We're going to talk about your intracellular buffer system and your blood buffer system. So the intracellular buffer system is phosphate, and where that comes from, it comes from ATP molecules. There you go. Okay, so the hydrolysis of ATP, remember, we need to get used to drawing an ATP molecule. So let's practice. Okay, so ATP is made up of 3 phosphates on a ribose ring and then with a nitrogenous base group, adenine. So if we start by drawing our 3 phosphates, remember they're phosphoanhydride bonds. So I just usually start by doing that. So PO, PO, PO. Off of all the phosphates, there is a double bond O. Coming down off of all of the phosphates is an oxygen with a negative charge. And then coming off to the left of the N phosphate, you'll have an oxygen that is protonated. Okay, this is going to be a common occurrence is that the linear oxygen on these phosphates, however it is, will have a proton on it, and the vertical oxygens are going to have a negative charge, and that's always if you orient the double bond to be pointing north. Okay. So what comes off of this terminal oxygen over here is we're going to have our ribose ring. So you have a methylene group here, a CH2, don't forget that guy. And then we need to draw a 5-membered sugar here. And coming off of this carbon, for now, you can just write AD for adenine. Soon, you will be drawing the structure of that nitrogenous base group though. Okay, so here is our backbone molecule of ATP. And like I said, you will need to be able to draw this for the exam. All right, so ATP is the currency of the cell. And we usually see a couple reactions. ATP can be broken down into ADP and free phosphate, or ATP could be broken down into AMP plus what's called inorganic pyrophosphate, which is 2. It's essentially you hydrolyze this bond right here. So you have these 2 phosphates linked together. But that inorganic pyrophosphate will be further broken down into 2 free phosphates. So this is where the phosphate is coming from inside your cells that's acting as that intracellular buffer. And now if we draw a free phosphate, you should use that idea. Oh, I'm on the edge there, sorry. You should use the idea that anything, I'll just move to the next slide. Anything in solution, free phosphate, you should see 2 hydrogens and 1 negatively charged. Now I always just draw it so that the double bond's pointing north and anything pointing south has a negative charge on it, and anything in that horizontal plane has the proton on it. But you know, this is just a little flipped around, but it still says the same thing. So it'll have 2 free hydrogens and a negative charge on it. Okay. Once inside the cell, this could potentially deprotonate. It will to some extent, and it'll give you this form of phosphate. So inside your cells, you've got these 2 species, these 2 chemical species acting as the buffering system inside your cells. I know. And if we look at some common nomenclatures for these, right? Full phosphate, we could call that shorthand H3A because it is a triprotic acid. So it has 3 protons on it, and the A just stands for acid. You could call this just the acid form. Once you deprotonate it once, right, our H3A becomes H2A minus, and you can start to call this the monobasic form. Okay, so every time you remove a hydrogen, that's going to be called the basic form. So monobasic means it's removed one proton. You can remove that second proton, and our shorthand name becomes Ha2 minus, and the way you could refer to that is the dibasic form. And lastly, you remove that very last proton and you have the A3 minus, and that would be called the tribasic form. So again, inside your cells, from the hydrolysis of ATP, you have both the monobasic and the dibasic forms present, and that's what's buffering all the intracellular reactions to remain at around a 7.6 pH. Titration curve, we should all be very comfortable looking at these. If we think about this is a triprotic acid, we notice we have those three horizontal regions, which are the buffering regions of it. There's three ranges, and that's always going to be, remember, plus or minus 1 pH unit from the pKa. So the three pKa values for phosphate are 2.2, 7.2, and 12.7. I told you that the intracellular pH hovers around closer to 7.6. So that would be on this chart, maybe around here-ish, right? Where I put this X. So what does that mean in terms of the species that are present in the cell? And remember, if we just recap titration curves, down here, it's 100% H3A. At the first endpoint, or the first equivalence point, you should have 100% H2A minus. At the second endpoint, you should have 100% HA2 minus. And then up here at the third endpoint, you'll have 100% A3 minus. So if we're stuck here on the titration curve, the only species, chemical species we have present are the H2A minus, so the monobasic form, or the HA2 minus, the dibasic form. Okay, and that's just what I had covered on the previous slide. So that concludes the buffering system of inside the cell, with the origin of it. So it's coming from the hydrolysis of ATP. And remember, you only have the monobasic and the dibasic forms present. Thinking back to the titration curve, since our pH intracellularly is slightly above the pKa, that means we have more of the base form, so the dibasic form, than we do the monobasic form. That could be a fun Henderson-Hasselbalch question. Tell me what the percent of, phosphate is inside the cell with a pH of 7.6. So you'd need to use the Henderson-Hasselbalch to solve for that ratio, A minus over HA, and you could tell me the amount of protonated or deprotonated form that's inside the cell at that pH. So moving on to the blood buffer system. The blood buffer system is not phosphate. So ATP is not really found outside of the cells. It's not floating around your body. But you do find, hold on one second. So you do find inside our blood, we have the bicarbonate buffering system. And this is because of the chemical reaction that occurs between carbon dioxide and water. So just to give you an idea of where all of this carbon dioxide is coming from, we have to think back to biology class, kind of intro biology, what Bio 212, maybe 214. And the carbon dioxide is coming through cellular respiration. So inside your cells, you're taking one molecule of glucose, you're burning it up in a combustion reaction, right? We breathe oxygen, it's necessary for us. And then that breaks down. Every one glucose molecule yields 6 carbon dioxide molecules and 6 water molecules. And we get some amount of energy, which is converted into ATP. Okay, but so every one sugar molecule that's broken down inside your cells, you're making 6 carbon dioxide molecules. The other route, or the other area where we're producing a lot of carbon dioxide, is beta oxidation. So this is the breakdown of fatty acids. So one fatty acid, and this is just stearate, which is C18, so stearic acid, plus 26 oxygens yields 18 carbon dioxide molecules, okay? So one yields 18, and I made energy capital here because beta oxidation produces way more ATP than glycolysis does. but we always just tend to be able to cover glycolysis the most. But if you're taking lipids, don't worry, we'll talk a lot about this beta oxidation. All right, so this is where the carbon dioxide is coming from. It's coming from the cellular respiration. So the breakdown of glucose to form energy and the breakdown of fatty acids to form energy. So how does this H2CO3, our carbonic acid, form? Okay, this is just a quick recap. So inside our cells, right? Sorry, I guess I'm finding the edge of the presentation here. Inside the cells, we've got the respiration going on, so we're creating a lot of carbon dioxide. So this is a cell. Okay, we've got our blood veins. So here we've got blood coming through, which is going to be providing oxygen, right, on hemoglobin. So our red blood cells are bringing lots of oxygen, and oxygen and carbon dioxide are going to freely how do we go back? I can only go forward. Okay, there we go. Are going to freely exchange. So there's lipid layers here. And remember, so they're hydrophobic and they don't want anything polar coming through the, I was going to draw now. They don't want anything polar coming through the membranes, but things that are non-polar can come through the membranes. So things that are non-polar are things like oxygen and carbon dioxide. Oh. So if we erase, there we go. Okay, that was a quick fix. So if we draw a molecule of oxygen, if we draw a molecule of oxygen, it's O2. Having technical difficulties. Ink. O. Annotate. We'll just use Zoom to annotate. All right, so oxygen is O2. Okay, and if we draw the structure of that, oxygen usually likes to make 2 bonds and have 2 lone pairs. The gases are actually really easy to draw if you know their bonding patterns. Hydrogen likes to make 1 bond, so H2 gas is just 2 hydrogens with a single bond. Oxygen likes to make 2 bonds and have 2 lone pairs, so oxygen gas is just 2 bonds with each molecule having 2 lone pairs. And then you've got carbon dioxide, which is the other gas we have here in play. Carbon likes to make 4 bonds, oxygen likes to make 2 bonds and have 2 lone pairs. So if we look at the electron distribution of these molecules, because that tells us whether or not things are polar, right? If we can draw dipoles, then we can determine whether or not a molecule is polar. So if we look at oxygen, for example, it's only 2 atoms and they have exactly the same electronegativity. So there is no net dipole at all on a molecule of oxygen. So it's non-polar and can easily move through the membrane. Non-polar. So those can freely dissociate off of the hemoglobin protein through all the cell layers and get into the cell so that you've got oxygen to fuel your cellular respiration. Okay. We look at carbon dioxide. Is carbon dioxide polar? Well, oxygen is more electronegative than carbon, so we could draw our dipoles to go this way. But carbon dioxide is also incredibly symmetrical. So even though we're tugging the electron density either way, those 2 vectors are going to cancel each other out. So overall, carbon dioxide has no net dipole and is therefore also nonpolar. So that just means that the carbon dioxide being produced inside your cells can easily, without any issue, traverse the membranes and make its way out into the blood as well. So once inside the blood, let's draw like a zoom up box here. Let me see if I can erase. Oh, okay, we'll just erase that. So once the carbon dioxide gets inside the blood, let's see what happens here. So we have a carbon dioxide molecule, okay? And inside the blood, we have lots of water molecules as well. And what we will see is the reaction between oxygen, water and carbon dioxide. So we've got, remember, one of the properties of water is that it's really nucleophilic, and we've got a partial positively charged carbon right here, because remember, those oxygens are more electronegative, so they're just ripping the electron density away. So that carbon is just feeling a little naked, a little exposed. And we've got this water molecule that has two lone pairs that are really high in electron density. So those lone pairs are going to attack that carbon, and one of these double bonds is going to break. It has to. Okay. So that would give you this kind of intermediate compound, O minus, with our water now attached. And this water, though, has a plus sign. Okay, so if you have a molecule like this won't exist for but femtoseconds, if even that, because we've got a positively charged oxygen on the left and a negatively charged oxygen on the right. So just pretty much immediately, you're going to see that the negative oxygen is going to rip a proton off that positive side over there. You can't have an acid and a base on the same molecule, right? This negative oxy anion over here is a base, and this OH2 plus on the carbon is an acid. So you just cannot have an acid and a base on the same molecule. They'll neutralize each other. So you net carbonic acid. And that's how the H2CO3 is formed inside the blood. This molecule is pretty unstable, so it readily dissociates. So it's going to lose an H plus, and you're going to get a lot more of this compound right here. So the monobasic form. Inside the blood, you'll see both the acid form and the monobasic form. Okay. If we move on to the next slide, I don't recall the pKs off the top of my head, but it's somewhere, I think it's 6.4. Let's go check. Let's check it out. Next slide. Stop annotation. Next. Okay. Nope. Next. All right. It's 6.4 exactly. So this is just another way you can look at and think of, it's not really a titration curve, but it's showing the same information. So on our y-axis over here, we've got percent total species in solution. So 100% and 0. Okay. And at the 50-50 point, right, if we're thinking about a titration curve, that's in the center of that horizontal spot. So at pH 0, we should have 100% of the acid form. That's the only thing we have in solution. As you keep increasing the pH, eventually it will start to deprotonate, and you'll see a rise, right, in the monobasic form. Okay. They'll reach some point where they're about 50-50. The monobasic form will peak at 100%. And then as that starts to decrease, you'll start to see the increase of the dibasic form, or the fully deprotonated form. Okay. So in the blood isn't 7.6 like the intercellular pH. The blood pH is about 7.4. So that's marked here on our x-axis, which is pH. So if we were to draw a line kind of straight up, we could see the percentages roughly of all the species present at pH 7.4. And it looks like it was maybe 25% acid and 75% monobasic. So that's how you would read this type of a curve. Okay. And I think, one last little kind of thought about the blood buffer system. So like I said, if you, so the cells are giving off lots of carbon dioxide, and it's moving into the blood. It's reacting with water to give you this carbonic acid, which readily deprotonates to some extent. So you've got these two species present. Okay. Once the blood travels to the lungs, the partial pressure of carbon dioxide in your lungs is less than that in the blood. So it's going to easily diffuse, again, across all the membranes and everything. So first it's got to go backwards through this and form carbon dioxide, but then it will readily diffuse into your lung space because the concentration of carbon dioxide in your lungs is less. So a question you can think about medically might be normally, here we'll start here. Normally, carbon dioxide is efficiently expired from the lungs. Patients with emphysema have impaired expiration. So what effect will that have on the blood pH? So they're not able to get all of that carbon dioxide out of their lungs. And what would be the reason for that? So that, to me, their cells are still working and making lots of carbon dioxide. So this side of the chemistry is still occurring, right? The problem that people with emphysema have is this side is not really being as efficient. So that's not really working. So what effect might that have on the pH? And you would look for, right, pH, don't forget, is the negative log of the concentration of the H plus, the amount of free protons in solution. So where do we have free protons here? On the right side of this chemical scheme, we have free protons being pumped into the solution. Okay, so things should be becoming more acidic. On the right side, when we run this reaction in reverse, when we go to like breathe out our carbon dioxide, we're absorbing the free protons from solution. And so that should make the pH become more basic. So somebody with impaired expiration should have what's called blood acidosis. That's where their pH of their blood is going to be less than the average person. So 7.4 is the average. Someone with blood acidosis might have a blood pH of 7.2, 7.1, 7, and that can wreak its own havoc, as we will soon learn with hemoglobin and efficient oxygen delivery. So that depends on the pH of your blood. So a whole slew of problems can happen if you can't efficiently get rid of the carbon dioxide that's being carried through your blood. So that wraps up the information about the two buffer systems of your body. So we talked about the intracellular buffer system, which is phosphate, and that comes from the hydrolysis of ATP. And then we talked about the blood buffer system, which is carbonic acid, which comes from the reaction between carbon dioxide, which is kind of a byproduct of cellular respiration, and water.